Judith will tell us about epigenetic programming of cell-phase decision, lessons from the immune and intestinal systems. Let's
Thank you very much for the invitation to come speak to you. So I'm standing before a class mainly of students, and I would like to give advice. No one has read the Advice for Young Scientists book. One should read it, but this is just my advice. If you are in the beginning of your career, make sure that you find great collaborators. And collaborators are students and also other PIs that collaborate. Because science is fun, but it's much more fun to do it together. And the way you do it together is you work with your students and you also collaborate with many other labs. So it makes your scientific life and your personal life very much worthwhile. So this is my title to my talk, and I'm going to start with lessons from the immune system. If we have time, we'll get to the intestine. So one of the dilemmas of molecular biology is monoallylic expression. And what I mean by that is how do cells choose between two alleles in a binary fashion. So it happens efficiently in each cell and every cell and randomly. So there are about 10% of the genes in our genomes are monoallylically expressed. And there are several types of monoallylic expressed genes. I'll just mention two of them. The imprinted genes, which are non-randomly expressed, they are expressed specifically from either the paternal or the maternal genome. And there are the random monoallylic expressed genes, which are expressed either from the paternal or from the maternal. In some cells, this is the paternal allele that is expressed. In other cells, the maternal allele is expressed. And the best known example for that is the X chromosomes, but also olfactory receptor, immunoglobulin, TCR, the T cell receptor, etc. So over many years, we have studied the, the immunoglobulin system in trying to understand how a lymphocyte chooses one allele for expression in each cell. And the immunoglobulin is a little bit more complicated because prior to expression, we have the rearrangement state. So in fact, the question is, how does rearrangement occur on one allele? And we know by now that it's programmed. It's not stochastic. It's epigenetically controlled. So as I show here, one allele is decorated with one sort of chromatin modification and DNA methylation, and the other allele is differently decorated. But one thing we appreciate already some time ago is that in addition to DNA methylation, chromatin, non-coding RNA, everything is found on the immunoglobulin loci. There is something else which is called asynchronous replication. What is asynchronous replication? So for most of the genome, which is divided into large zones that replicate according to a fixed program, some replicate early during S, others in the middle S, and the others in the late S. But in all cases, the two alleles replicate more or less at the same time in S. However, there is asynchronous replication. And by that I mean that this is one way we measure asynchronous replication is by FISH, simple DNA FISH, not so simple, but simple. And we have probes for the regions that we are looking at. So at the beginning of the cell cycle, at G1, we have two single dots, which means the two alleles have not replicated yet. At the end, at G2, we have two double dots. Both alleles have replicated already. And in S, we have single, single, double, double, but we also have this population of single, double. So the single dot represents the allele that has not replicated yet. The double dot represents the alleles that has replicated already. And we call it single, double, and the percentage of single, double nuclei that we see represent if a locus replicate in a synchronous or in an asynchronous manner. Exactly. We don't, I mean, the way we do it is we just label if we want to look only on S-phase cells. Okay? So we label this way with BRBU, so we know the replicating cells. So some time ago we showed that for immunoglobulin and other genes, we have asynchronous replication. In one cell, the paternal allele is early and the maternal is late. In the other cell, the maternal is early and the paternal is late. And then we looked what happened in a single cell clone, and you will understand why down the line. Here, the mother cell is paternal early and maternal late. And now we look at the other cells in these clones, and all of them show the same pattern: paternal early, maternal late. So this replication mode, the asynchronous replication mode, is committed. The cells are committed to one mode, either one or the other, but once they make the decision to replicate, let's say, the paternal early, they will keep on doing it. So we concluded that every somatic cell type that we have checked shows a pattern of asynchronous replication, and this provided us with a hint that the decision to replicate asynchronously occurs very early during development. So we looked at the development. Yes?
Sorry, the last slide. So how about the different subtypes of the cells?
I can't hear you.
How about different subtypes of the cells? The B cells is different subtypes, so do they also target the replication time?
Yes, we have checked that in the B cell compartment mostly, and we know exactly when they start to be committed to this replication. Some cells are committed paternal early, some cells are committed to maternal early. So we looked during development, and we showed that at the beginning, in the blastocyst, we have synchronous replication. But right after implantation and from then on to the life of the animal, these particular regions replicate in an asynchronous fashion. But what happens in ES cells, a progenitor cell, or in this case, embryonic stem cell? So the mother cell replicates the paternal early and the maternal late, and then we made a single cell clone from them. And now we got that 50% of the cells replicate the same way that the mother cell replicated, but 50% replicated the maternal early and the paternal late. So it's different than the committed pre-B cell. In ES cells, they switch their decision. So when it's synchronously replicating, then they replicate at the same time, the paternal and the maternal allele. In B cells or in somatic cell, you have a commitment, either the paternal early or the maternal early, and they stay the same. But in ES cells and other stem cells like hematopoietic stem cells, neuroprogenitor, etc., we have this switching mode. At one time, the paternal allele is the early, but once the cell divides, the maternal allele is early. So here I show you one experiment that we did to show that the switching is a decision that is a chromosomal decision, and you don't need other chromosomes to remind it what to do. So this is a haploid ES cells, and in the middle we found a locus that replicate around synchronously and around the middle of the cell. Then we have this asynchronous region. So if we get this kind of picture, that we have double dot for our control and single dot for our test, we say that it's late replicating because it still has not replicated, although the control replicated. But if we get this picture that we have double-double, it means that the asynchronous replicating region replicates prior to the control. So we scored this phenotype, and you can see for two types of loci that replicate asynchronously, that about 50% are early replicating and 50% are late replicating, and we had the right control, those that replicate early and those that replicate late, and in printing that doesn't change. So indeed, we decide that switching is built into the chromosome themselves, and you don't need communication between the two alleles. They will switch each chromosome on its own. So why do we think switching occurs? We don't know, but we think it provides a form of plasticity to ensure that all tissue derivatives will have the cells that retain both possible options for allele specificity. So this is a summary of the decision points during the asynchronous process. So we start with the gametes, and we think that the marking of the chromosome, which one replicates early and which replicates late, occurs in the gametes. Then we have the blastocyst cells, in which I showed you that they replicate synchronously. We have the initiation of asynchronous replication, the choosing, the first time they choose which allele is early. Then we have in stem cells the switching from one allele to the other. And then in differentiated cell, we have the commitment. They have committed to one allele to being early. So by doing FISH, we were able to look at 6, 7, 10 probes, but we wanted to map many of the asynchronous replicating regions in the genome. And for that, what we did is we took an F1 mouse between Castaneus and B6, purified bone marrow cells, grow single cell clones from each of them, prepare the DNA, and then sequence G1 versus S. So B6 versus Castaneus, they have SNPs. So once we get all the reads, we divide the reads according to alleles, and we also calculate the replication time if we have a change in the ratio of the counts that we get in S versus G1. And this is the map that we get. So for each one, for each cell that we do, or clone that we do, we have 3 replicates. And we look at the 2 alleles. The B6 is the red one, the Castaneus is the blue one, And let's look at the enlargement, and I would like to point this area, which is marked by red, which means that the B6 allele replicates prior to the castaneous. And here in blue is the opposite, castaneous prior to the B6. And I don't mean to ask you to read it, but we validated, we got a lot of novel asynchronous replicating regions from this whole genome study. We validated 16 of them in fish, but even more importantly, we showed that this is what happens not only in the pre-B cells that we are checking, but also in ES cells, in MEFs, and even in a human clone. So what are the characteristics of these regions? They replicate relatively late in S. They are enriched in lines, not in sites. They are enriched for the B compartment, LADs, and low expressing genes. They are enriched for monoallelic transcription units, and the early replicating allele is in a more accessible chromatin, as we have done ATAC to analyze it. So this is just one example. This is the replication. So B6 replicate prior to the castaneous. Transcription is coming from the B6 allele. No transcription is coming from the castaneous allele. And then ATAC peaks are coming from the B6 and not from the castaneous, and vice versa for the castaneous early. So what's special about these regions? So in addition to asynchronous replicating regions that we know, like immunoglobulin, NK receptor, TCR, olfactory, we also saw a sensory perception of taste, G protein coupled receptor signaling, sensory perception of smell, again, etc., and also chemataxis and motility that no one has known before. So are all asynchronous replicating regions on any given chromosome replicate in the same orientation? There was a publication that checked only 6 probes that saw that along the chromosome, all the asynchronous replicating regions are either early or late. So now we had many more, like close to 500, and we wanted to see if this is true. And this is absolutely not true, because as you can see here, I marked in red the B6 early, in castaneous the castaneous early, and for almost every chromosome you see the asynchronous replicating regions. Some of them are early on B6, others are early on castaneous. So then we ask, is there a fixed relationship between asynchronous replicating region on any given chromosome? So for that, we looked for parallel and anti-parallel coordination. So what do I call parallel coordination? So parallel coordination is that when we take 2 probes, one red and one green, and we do a fish, and you can see that the double dot of the red is next to the double dot of the yellow, green, and the singles are together. This is one chromosome, this is the other chromosome, so they are in a parallel orientation. They behave the same. Anti-parallel orientation is that we have a single from the red and double from the green. So on one chromosome, they replicate in an anti-parallel coordination. One is paternal early, the other one is maternal early. So doing this type of experiments on different chromosomes, so we have 3 different chromosomes, and then different cell types, we analyze what I show here is the percentage of the anti-parallel. So let's look at that. A and B are anti-parallel because A is B6 early, B is castaneous early. And around 80% of the nuclei that we analyze behave in a parallel fashion. A and C are parallel, both are red. So how many anti-parallel coordination we find is about 27, 17, 26, and so on. So yes, there is a fixed program. So there is a fixed relationship between asynchronous replication and any tested chromosome, and both parallel and anti-parallel exist. So if we think this way, then we probably should conclude that we have two types of cells, type 1 and type 2. In type 2, probes A and C are early replicating, and they are in a parallel coordination from the paternal allele. In type 2 cell, A and C are still coordinated, but they are late on the paternal allele, and vice versa. So we think that all cells are in either one of these orientation, because there is a fixed relationship between asynchronous replicating region on any chromosome. And probably it is set up early in the embryogenesis, and most probably on the ground of marking, most probably in the gametes. So to thank the people, my great students that did this work, and our collaborator. So you see here Fernando, he likes to be called Fer, and Marganit is not here, and Lamia is here. So Lamia and Fer, please raise your hands so people will know how to ask you a question. And my all-time collaborator and friend, Howard Cedar. And for the genome-wide, we teamed up with Itamar Simon from our faculty, and most of the computation work that I showed you until now was done by Brittany Blumenfeld, his PhD student. So if you have questions to this part, maybe you would like to ask them, or yes.
The replication plots you show early to late, so they're inferred from just sorting G1 versus G2. So I'm not really sure how you do that. If you have two populations, how can you then infer the whole S-phase progression?
We count, every region is counted, okay? And we calculate the ratio. And then we do the SNPs. Maybe I can go back and it's easier to explain.
Red and blue, yeah.
Yeah, no, but I wanted to get to here. So these are the way we do it. We first get the SNPs. Then we did realign the SNPs, and then we divide the alleles according to the SNPs. And then we calculate for each position the S to G1 at a resolution of 100 kilobase, okay? And then we had a statistical method that will allow us to tell whether the difference that we see are significant. So we know that we probably, due to this very harsh statistical test, we probably lost asynchronous replicating gene. We also did it on several clones and combined, and yeah, so it went through a very rigorous testing, and then testing in fish to know that we really are looking at what we are thinking we are looking at. Any more questions? Yes. What is the situation in? Okay, we didn't check the trough ectoderm cells, so I cannot tell you. Right, Lamia? Right, Fer? But it's a good question because the imprinted genes are already replicating there, we know, in one direction. I don't know.
May I ask a question? First, did you exclude genes that are affected by strain?
Yes, that's a very good question. Yeah, before, that's true. We excluded all areas that are affected by, that we can see them in the paternal, in the, in one strain and not in the other. Yeah, this is very important. This is not gene. This is not sequence related. Very good.
Okay, my second question is, why do you think we don't have a problem if when, okay, in the ES, if the two chromosomes don't communicate one with another one, why we don't have problems in the cell?
So you're asking, since switching occurs independently, each chromosome is switching on its own. It doesn't need to ask his partner. And we think that it's probably all dependent on the initiation step. So I think they are marked priorly. So the true answer, we don't know what causes the switch. It can be either in cis or in trans, so we don't know. But you don't have to choose every time you replicate, because we believe that the mark was put on early on and you just see it now. So the mark is probably from the gametes, and that tells for ESL, or the blastocysts that are going into implantation, to know whether it's early or late, and then it just like changes every time. How? I don't know. But it doesn't, I mean, they survive. Not only they, hematopoietic stem cells, MMPs in the hematopoietic stem cells, mammary progenitors, and so on. Okay, so I'll go now to the second part of the talk, which lessons from intestinal system. So some years ago, we became interested about on the processes of inflammation. Why? Because we figured out that, like other people, that is linked to the pathogenesis of tumors in substantial fraction of human cancers, and it plays a causal role in cancer development. So then why did we choose the IBD, the inflammatory bowel disease? Because the incidence of this disease is increasing among adults. And also we knew that about 10% of IBD is heritable, attributed to genetic variants, but the 90 other percent are not. And we are interested in epigenetics, and the intestinal tract really records very well the environmental challenge put on the cells due to the close contact with everything that goes through the intestine. So this is the colon, because the first part from this, it goes with the colon. These are the crypts. Colons don't have villi. And if we go to the bottom of the crypt, we see the stem cells. Then we see the trans-amplifying cells. Then we see some goblet cells. Then we see some tuft cells. Then we see enteroendocrine, and there we see the enterocytes. So we asked the question, what is the role of microbiota-induced epigenetic mechanism in modulating homeostasis and gastrointestinal pathology? So first, we compare GF is germ-free mice who are grown in isolators, no contact with microbiota, to CNV, conventional raised mice, and we did RNA-seq and whole genome bisulfate. If we look at the RNA-seq result, the difference between GF and CNV is only in DNA replication, in S phase. Please remember that. And then we said, okay, we are epigeneticists, let's look through the eyeglasses of epigenetics. We did whole genome bisulfate. And at this particular study, we concentrated on LMRs. These are low methylated regions, identified first by Dirk Schubler, and they are putative enhancer elements. They are conserved in evolution and enriched in active chromatin marks, depleted in repressive chromatin marks, contain many transcription factors, binding site. They look like enhancers. So we characterize the LMRs that we get in CNV versus the GF, and we saw that we have a lot of LMRs that became hypomethylated in CNV versus GF. And then we overlapped the data that we have from the RNA-seq and the whole genome bisulfate, and we concentrated mostly on the hypomethylated LMRs that were linked to genes that are upregulated in expression, simply because this is a more direct interaction that we can understand better. Yes?
Are these LMRs producing specifics along the colon?
If they are what?
Because you can find more LMRs at the beginning or at the end of the colon.
So we didn't do that type of analysis. We are thinking of doing it right now, but yeah, I don't know. We just took the crits, all the crits, and we validate everything that you see by FAC sorting intestinal epithelial cells. So as I said, we concentrate on this group, which is the hypomethylated LMR with genes that are upregulated. We ask, what do we get there? So we were sort of surprised at the beginning that we say that we get colitis and an inflammatory bowel disease. I want to stress for you that we compare GF to CNV. No inflammation whatsoever. And still we get colitis and inflammatory bowel disease. But then when we look inside to see which type of genes we get, we see that this set of genes are enriched for genes that are antibacterial and anti-inflammatory response. So we call this gene, the set of genes, early sentinel inflammatory genes. So we conclude that exposure to commensal microbiota leads to transcription activation of early sentinel inflammatory genes, which are probably important for normal intestinal homeostasis. And this insight was gained from methylation analysis on top of the RNA-seq, because RNA-seq, as you remember, just gave us DNA replication. And it emphasized
sequencing studies, both trying to identify germline variants that predispose to develop certain types of cancer, but also somatic mutations that occur with a particular frequency in non-coding parts of the genome. And in this figure taken from a paper published now some 7 years ago, you can see that there's a couple of mutations that occur in the promoter of the third gene with a very high frequency. So in particular cancer types such as melanoma, these promoter mutations occur in almost 50% of the cases. So it's a very frequent and very important driver event, these mutations in the promoter of the third gene. So that's provided the first clues of what this transcriptional reactivation of third, what the molecular mechanism is, because it may involve these promoter mutations. So then mass spec comes into the picture, because we have technology that allows us to study the functional consequences of these mutations, essentially addressing which proteins interact with the wild type third promoter compared to these mutated sequence. And we use quantitative mass spec for that. So the principle is shown over here. it's very, in principle, very simple. We take cells that we grow in culture. In this case, this is a melanoma cell line that you can grow in the lab. We make nuclear extracts, and these extracts are then used for in vitro DNA pull-down experiments. So you use the wild-type DNA sequence with a biotin on it, so you can immobilize it on streptavidin conjugated beads, and we fish with the wild-type DNA sequence and the mutated sequence in extracts derived from these melanoma cells. After incubation and washes, all the bound proteins are digested with trypsin and then differentially labeled with, in this case, a dimethyl label, so we can discriminate the signals from the wild-type pull-down and the mutant pull-down in the mass spec with a distinct mass shift. So you get in the mass spec always doublets, a signal that corresponds to a peptide signal coming from the pull-down with the wild-type sequence, and a signal that comes from the pull-down with the mutated sequence. And background proteins would then show a 1-to-1 ratio, so they interact equally well with the wild-type and the mutant sequence, or they bind non-specifically to the beads. But if there's a protein or transcription factor that specifically interacts with the mutant third promoter, you get a higher intensity in the pull-down with the mutated sequence compared to the wild-type control. So this then indicates that this protein here, this peptide and the protein this peptide belongs to, is specifically interacting with the DNA sequence when this base mutation is introduced. So we also perform a label swap, and then you can visualize in a two-dimensional graph all the ratios of the proteins in this experiment, and also when we perform a label swap, and then mutant-specific interactions are visualized and in a quadrant like this. So we can very easily distinguish background proteins from the mutation-specific transcription factors.
What is the size of the DNA?
So in this case, we usually make use of about 30 to 50 base pair sequences with the mutation smack in the middle of the DNA sequence. So you can just order those at a company, and it's very easy. So just to remind you, this is the coding sequence of the third promoter. So this is the third gene, this is the gene here. This is the promoter sequence, and here you see these mutations that occur in cancer. And in green you can see these are transcription factor motifs. Upon introducing these mutations, you introduce a perfect binding site for ETS transcription factors. So the hypothesis based on this analysis is that upon introducing these mutations, you reconstitute an ETS transcription factor binding site, and that then would result in increased binding of these ETS transcription factors, and they then would be able to drive expression of the third gene. That would be the mechanism. But there are around 20 ETS transcription factors in the human genome. So we then use quantitative interaction proteomics to determine which of these ETS transcription factors is responsible. So this is the two-dimensional graph that we generated with the ratio versus ratio plot. The background proteins, this is typically around 1,200 proteins or so that we sequence in the mass spec, but they're all background proteins. And you see a small cluster of proteins that have a binding preference for the DNA sequence when it carries this mutation. And reassuringly for us, all of the proteins that we see here are all ETS transcription factors, but we still see quite a number of them in this quadrant over here. So the question then is, which of these factors shown here is really the one that binds to the mutant sequence and drives expression of TERT? So in order to investigate that, we performed perturbation experiments. So we knocked down this JBPA transcription factor and the ELFs here that you see here. And in the middle you can see if we knock down ELF1 and ELF2 in melanoma cells, you can see that by siRNA we can strongly reduce the expression of these two transcription factors, but we only see a very modest effect on TERT expression, the black bar over here. But if we knock down this guy here, JBPA, you see here the knockdown for JBPA, we get a very strong reduction in TERT expression in these cells. So using quantitative mass spec and these knockdown experiments, we've then identified that it's this transcription factor JBPA that upon introducing these base mutations here, that serves as a high affinity binding site for JBPA, and this molecular event then contributes to TERT expression reactivation in cancer. So this is just one example study of how we are using quantitative interaction proteomics to uncover the functional consequences of non-coding mutations in cancer. That's summarized over here. This JBPA is the transcription factor that selectively binds to and activates the mutant TERT promoter in melanoma. And in more general terms, quantitative interaction proteomics is a very powerful tool for functional cancer genomics and in post-GWAS studies. And we're using that technology in a very broad scale across many different cancer types and many different studies. You can look up these publications. So that was the first part. Oh, this is missing the fourth one. But so the next topic is this one here, a new mass spectrometry-based method that we developed to determine affinities between proteins and DNA sequences of interest. Now I'll show you this plot that I just showed you a few minutes ago again. These types of interaction plots, they are very strong tools to determine binding specificities, right? So we know that all these proteins labeled in red here bind with a higher affinity to the mutant third promoter here compared to the wild type sequence. But we don't get a readout about the actual affinities. So what is the binding strength between these transcription factors and the DNA sequence? And it's not only the specificity, but this binding affinity is also particularly relevant, and this determines whether in vitro determined binding events, whether they're also relevant in vivo. So we wanted to see if we can develop a mass spectrometry-based methods that allows us to identify proteins, but also quantify apparent affinities between these proteins and these DNA sequences. And in the biochemical, protein biochemistry community, there's various methods you can use for binding affinity quantification between proteins and DNA. You have ITC, and the experiment I'm going to talk to you a little bit about is EMSA, so electromobility shift assay. So this is the principle of that assay. The classical version of the assay makes use of radio-labeled, so radioactive DNA, and you have a certain concentration of DNA that is radio-labeled, and then you titrate in different reaction tubes a transcription factor that you're interested in this case it's FOXA1 or the DNA binding domain of FOXA1. And you start off with no transcription factor, and then in these multiple reactions you gradually increase the concentration of the purified transcription factor, and at some point, depending on the KD between the DNA sequence and the transcription factor of interest, you get binding in solution. And this is visualized in a non-denaturing gel, where a shift in the gel implies that there is now transcription factor binding to the DNA. So then you increase the concentration further. At some point, all of the DNA is bound by the transcription factor. So based on these experiments with the titration, in this case of the transcription factor, you can determine a hill-like curve from which you can determine a dissociation constant. Now we wanted to develop a mass spectrometry-based method based on this principle that allows us to determine multiple of these hill-like curves in an unbiased manner in the context of crude lysates and not making use of purified single proteins. And the method that we developed for that is called PACBAN, or Protein Nucleic Acid Affinity Quantification by Mass Spectrometry in Nuclear Extract. And the principle is outlined on this slide over here. I'm going to talk you through it. So we start off by generating nuclear extracts from cells that we can culture in a culture dish in large quantities. And then we prepare a titration series, not of protein, but of DNA. So in a KD determination, one of the two parameters has to be titrated. It doesn't have to be the DNA. It doesn't have to be the protein. It can also be the DNA. So we prepare a concentration range of the DNA containing a transcription factor binding sequence that we're interested in, from the low nanomolar concentration up to a few micromolar. We have 10 titration points. And this concentration range of DNA is immobilized on a 96 well plate. And these 10 reactions are then incubated with a fixed amount of nuclear extract. After incubation, we wash away the non-bound proteins, and then all these proteins that are bound to the DNA of the 10 reactions is digested into peptides. And these are then labeled with a mass tag that allows us to quantify the relative binding of proteins in these 10 reactions using quantitative mass spec. So in the mass spec, we then see reporter ions appearing from the 10 reactions, and based on this, we can then determine hill-like curves. But now we're generating dozens or hundreds of these curves in a single experiment, making use of nuclear extracts and using a concentration range of DNA. So this works really well. And this is all published, so I'm going to show you just a couple of example figures from this work. So here we took the SP1 motif, one of the most well-known mammalian transcription factor motif. There's various proteins that are known to bind with SP1 motifs. Here you see some of them, and we get a very nice range of affinities between the DNA and these transcription factors. So we really get KD curves that look very nice, that are reproducible, and they are sort of ranging between low and high affinity. Yeah, go ahead.
Do they correlate with the results from literature?
Yes. I come back to that in a minute, I think, a little bit, yeah. But I have to emphasize that what we see with crude lysates in these experiments is not necessarily always perfectly reflected by recombinant expression. So you can imagine if there's two proteins that as recombinant proteins would show a very similar affinity, but in vivo, in the cell that you work with, if one of them is expressed 1,000-fold more than the other, you can imagine that there's, at the lowest concentration range, some competition between the high abundant protein and the low abundant protein, and that you get apparently an apparent affinity for the protein that is more abundant. It would seem to have a higher affinity compared to the other protein, right? But these apparent values are still, I think, biologically more meaningful than results that you obtain with recombinant proteins, because the expression that you see in the lysate is the expression level that those transcriptions have in vivo, right? And this competition is naturally also occurring. But that's a very good point that you bring up there. And here you see that abundances do not dominate these readouts. This is, again, coming back to your point. Here's two replicates. And here you can see biological replicates. So we get very good quantitative data between two biological replicate experiments. And we can use mass spectrometry also to, in absolute terms, quantify the abundances of these proteins in the lysate using absolute quantification. And we did that for these cells that we use for these experiments. And the size of the dot here represents the abundance of the proteins in the lysates. And you can see that small and large dots are basically intermingled, which you would expect, because you want these measurements to be determined by the affinities and not by the abundances of the proteins in the lysates. So this is some orthogonal validation that we did. You can make use of a database called Jasper, which for every protein in the human genome makes a prediction or a motif score that tells you what is the probability that this particular protein interacts with this particular motif. And we see a very nice correlation between the Pacman measurements and the Jasper motif score for these proteins, in the sense that if we get a very high score for a motif in the Jasper database, we measure a very high affinity. And for proteins that do not give a very good score in the Jasper database, we get actually a low affinity. Finally, concerning specificity, we have to do additional experiments because we don't make use of a wild type and a mutant sequence in these experiments. We just probe, in this Pacman experiment, the wild type sequence. But if we make mutants, if we introduce a base pair mutation in the KLF motif, you can see, at least by pull-down Weston, that we completely abolish the binding between these three factors that are quantified here. Here you see the signal with the wild type sequence, and a mutation in the sequence completely abolishes the interaction. And we're working on a second version of this experiment now, where we include both information about specificity and affinity in one experimental setup, but that's not finished yet. So an outlook and a summary of this second part. So we developed a method that can be used to determine protein nucleic acid affinities in the context of crude lysates. And this is very exciting for us, because this, in a systems biology context, connects transcription factor abundance, which I told you we can now measure, and affinity of binding, which we can also measure to transcriptional output. So a combination of measuring the abundances of these transcription factors in cells and the affinities allows us to make much better predictions which transcription factor in vitro is binding to a particular sequence in vivo. I showed you a couple of examples of protein DNA interactions, but we actually also use Pacman to quantify affinities with reconstituted nucleosomes that carry certain modifications. We're using it for RNA modification. So everything that you can quantify and immobilize on beads, you can use as input material to measure these affinities. We are working on a methodology. Actually, the proof of principle of this is already working, where we combine different quantification strategies to combine 20 affinity purification into one measurement that allows us to get information about specificity and affinity. And what we're very excited about is that we continue our collaborations with Till Bartke in Munich, who's building reconstituted nucleosomes by introducing DNA elements in and in between nucleosomes, together with epigenetic modifications, to ask how affinities of transcription factors for nucleosomal substrates carrying particular transcription factor motif is affecting affinities with these substrates. Okay. I'm going quite fast, am I not? I should slow down a little bit, perhaps. Okay. If there's any questions in between, go ahead. Yeah, that's a very good point. So phosphorylations are usually substoichiometric. So in principle, when you have this data, you can search your mass spec data for evidence for any post-translational modifications on those transcription factors that may regulate its affinity. We haven't really done that much because without an enrichment for phosphorylation, it's very difficult to map those using mass spec. But you can think about all sorts, because you work with crude lysates, you can, for example, one of the things we're doing is to work with retinoic acid response nuclear receptor elements, where you can switch between repressive and activating receptors by hormone ligand. And you can do Pac-Man experiment with hormone-treated cells, for example, versus control cells, and see what your Pac-Man experiments, which affinities are maybe differentially regulated and so on. So that's the nice thing about being able to work with crude lysates, that you can do stimulus or any type of treatment to the cells and then use lysates from those cells to do these experiments with, which is actually something we're also doing at the moment.
I was wondering about...
I'm not done yet, but
I was wondering if you can do cooperative binding on a longer oligo.
Yeah, that's actually very interesting, and I actually show you in the last part of my talk that there's plenty of evidence for this cooperativity that we can see in our data. And so in principle, you can make use of an as long fragment as you want. We've used nested PCR to amplify enhancer sequences and used this as input material. So these things are all possible.
And also, have you tried to compare naked DNA and nucleosomal DNA in terms of the affinity to particular factors?
We haven't done that yet. So the nucleosome experiments that we did were just with two 601 dinucleosomes. We haven't really played around with linkers, and this is things that are... So the bottleneck there is TIL. So it takes him a long time to build and reconstitute those nucleosomes. So that's so he's the bottleneck, not us. But this is this hardcore protein biochemistry that takes time. So, but we're working on this. Now, in the third part of my talk, I'm going to show you how we are making use of these types of technologies to gain further insights into gene regulation in mammalian cells. So this is a collaborative study that we recently published with the lab of Dirk Schubler. So this work stemmed from a paper published a couple of years ago in Nature Biotech, in which they make use of massive parallel reporter assays. And what they do here is that they basically assay in a large-scale study the autonomous ability of DNA sequences in the human genome to drive expression of transcription by hooking up these DNA sequences to a luciferase reporter. And you can do this now in large scale, so you can assay a large amount of DNA elements in these types of experiments, making use of libraries of reporter constructs. And in this data picture that you see from this paper here, you can see each data point here represents, I think, a subset of DNA elements that carry particular motifs. And you can see that this is basically a score, so if you get a high score, this means that these DNA elements are positively correlated with gene expression. And here you can see a negative score implies that a DNA element is associated with silencing of transcription. And on this axis, you can see transcription factor binding sites for which we know what the transcription factors are that regulate expression from these DNA sequences, such as here the ETS motif, NRF1, and so on and so forth. They also use two types of cells for these experiments, HepG2, which is a liver cell line, and a hematopoietic cell line. And you also see cell type specificity here, because here you see, for example, HNF4, which is a transcription factor that is expressed in liver cells but not in hematopoietic cells. So you only get a score for these elements in the liver cells. And you also see here GATA transcription factors which are expressed in hematopoietic cells. So these elements only get a positive score in the hematopoietic cells but not in the liver cells. Now, in this study, there was something very interesting. There's a motif, which you see the sequence here. It contains two CGs in the middle of the motif, and it very potently activates transcription. However, the transcription factor that is known to interact with this motif was unknown. So this is a perfect case study where quantitative interaction proteomics potentially is very powerful to address this question and ask, what is the transcription factor that interacts with this motif? Something about this motif, it's very common in Cpg island promoters in mammalian cells. As I said, it's strongly associated with gene activation, but the transcription factor that interacts with the motif is unknown. So we thought this was an interesting case to go after. So this is data from the Schubler lab, recapitulating what is already known. If you put this motif in front of a luciferase gene. This is now a reporter assay in mouse CS cells. You can see a very dose-dependent and very potent activation of transcription by having 1, 2, or 3 motifs in front of a luciferase gene. So we then performed quantitative interaction proteomics. So first we performed the specificity experiment with the wild type and a scrambled motif. And in this quadrant here, you can see the proteins we identify as having a binding preference for the CG motif compared to a scrambled motif. And 2 proteins stood out, BAY and P and BEND3. So these are both BEN domain containing proteins, one of which is known to be a transcription factor. The other one was completely not characterized as a transcription factor. So we were at first a little bit puzzled about this, also because BEND3 had been implicated in regulating satellite repeat repression and so on. So it was a confusing result, and we didn't really know what to think of it. But it was interesting enough to also then pursue with a Pac-Man experiment. And that was very interesting, and it shows you why these Pac-Man experiments give us more information than the specificity experiment does. Because in Pac-Man, one of these 2 proteins, BEMP, yielded a very high affinity in the low nanomolar range, whereas this BEND3 protein was not quantified with an affinity. So even though both of these proteins in the specificity plot are identified as specific interaction partners for this particular motif, the affinity measurement told us that this is the guy that we're really after, but this one isn't. So BAY and P, is this a transcription factor? Is this the one that's responsible for driving expression from this motif? We were still confused because I summarize here a little bit the knowledge that is known about this protein. So it's a BEN domain containing protein. It's in the center of the protein. It's a small or average sized protein. It's also known as SMAR1, supposed to bind to the nuclear matrix and silence genes. It's not annotated as a transcription factor. It's an essential gene, so you cannot live without it. BEND3, which is the family member of this, recruits PRC2 to major satellites and fly BEN protein function as repressors. So if you were to find this protein in your mass spec and do a literature search, then you would be a little bit discouraged and thinking, I must be wrong, right? This can't be right. This can't be a potent transcriptional activator. But the Pac-Man experiment was so clear and the protein showed such a high affinity for that motif that we continued to study this protein functionally. So we performed chip sequencing using an antibody against endogenous BEMP, which was luckily for us available. And so here we have 2 replicate ChIP-seq experiments, and reassuringly we could see that the BEMP ChIP-seq revealed binding at transcription start sites, and the motif uncovered underneath the ChIP-seq is exactly the motif that we used in the DNA pull-down. So in vivo, this protein is indeed binding to this particular motif on Cpg Island promoters. So that was reassuring. The CG motif in Cpg Island, of course, cytosins are known to be methylated. So we also investigated what the consequences are for binding of BEMP to the motif if we methylate the cytosins here, and that results in a strong reduction of binding affinity. Here you see the affinity for the wild type sequence, and this is when both of these cytosins are methylated. So methylation seems to be a regulatory switch that determines whether BEMP is able to interact with these sequences or not. That's also supported by additional data that I'm not showing, namely that there's mouse cells that are deficient for DNA methylation, so triple DNMT knockout cells, and we see vastly increased binding of BEMP in the genome of these mouse ES cells compared to wild type ES cells. And also in human cancer cell lines, we really can see that BEMP binding in the genome is following DNA methylation, meaning if a certain locus is methylated, we don't see binding of BEMP, but if it's unmethylated, we do. So DNA methylation seems to be a regulatory mechanism that switches the binding of BEMP to these Cpg Island promoters. So to functionally study the protein, we made use of the DTEC Dagron system developed a couple of years ago, which allows you to acutely deplete a protein of interest very rapidly. So here you see a Dagron line, which also, together with the Dagron, we knock in a V5 tag in the locus, and in a couple of hours you get very nice complete depletion of BEMP. This is important because, as I told you, cells can't live without BEMP, so you can't make a CRISPR knockout. But this allowed us to at least study functionally the protein further in ES cells. So here you see some data we got from the Dagron experiment. So here you see a binding site for BAMP, a particular site in the genome promoter that's bound by BAMP. And we can see by RNA-seq that if we activate the DTAC, we see a very rapid reduction of transcription from this particular target gene. We first see intronic RNA going down and then also exonic RNA functions, indicating that it's really a direct target of BAMP binding to these promoters that results in transcription, and if you remove it, you get reduction of transcription. And this is also at some point followed by a reduction of the protein that is encoded by the target gene after the DTAC treatment. So from these DTAC-based experiments combined with RNA sequencing, we could conclude that indeed this BAMP protein, this peculiar protein that we never expected to be a transcriptional activator, it binds to and potently activates the expression of essential genes. And this is also an important point. The RNA sequencing revealed that BAMP binds to important housekeeping genes and regulates its expression, which also explains why cells can't live without this protein when you knock it out. So the summary of the third part is that we identified BAMP as the long sought after transcription factor that interacts with an orphan CpG motif that is very common in mammalian Cpg island promoters. DNA methylation strongly inhibits binding of BAMP to this motif. And BAMP is a previously unknown transcription factor that strongly activates housekeeping genes, positions nucleosomes, and regulates chromatin accessibility. These last two things I didn't have time to show you. And I think, yeah. No, we haven't done that yet, but that's an interesting question to look into. Yeah.
You spoke about another protein that you found and that the affinity was lower. Maybe methylated DNA is the same.
Yeah, so we and others are looking.
And so I think we're now moving to the very last and surely not least lecture of this school for epigenetics and stem cells, and I'm really honored to invite Professor Howard Cedar, which I would say the father of epigenetics, and he will give a talk about mapping the epigenetic flowchart of development. So we're really excited to hear your talk.
Okay, and thank you all for staying around. And I'd just like to, there'll be no surprises here. I'm going to talk about DNA methylation. And what I'd like to do, I think I could just yell, it's fine.
No, but they can't hear you at all.
Okay, I'm sorry. Well, everything in life is complicated these days. Okay, is that good? Can you hear me? So we have to turn this on. Now you can hear me? Okay, great. Okay, so let's just start off in a simple manner. I don't know which one. This is the. Oh, good. So I'd just like to give you the overall picture of what DNA methylation is, without any of the details and any of the biology. And basically, it's an annotation system. In other words, you have a genetic text, which you all are very, very well familiar with its letters and words and sentences. But in addition is DNA methylation. DNA methylation, let's see. How do I get this to move? Or doesn't? Okay, and that's DNA methylation, which is only on CpGs. And this methylation is basically, it doesn't change the text. You can also change the methylation all the time. It doesn't change the text at all. The text remains the same. But DNA methylation is used in the cell as an annotation to tell the cell how to use the genetic material. Okay, and this is used during development, it's used after development, it's used in disease, it's used in all sorts of biological situations in the cell to alter the activity of genes in the cell without changing the gene, the text itself. I'd just like to, because we're here in Jerusalem, I'd just like to show you this fantastic example. Many languages have methods of annotation, right? We all put emphasis on letters, we all put lines over letters, we all put all sorts of annotations on our text. But the best example, I think, in the world is also probably one of the oldest languages, and that's Hebrew. And what you see here, this is a copy from the Damascus Codex, which is a copy of the Bible that contains the text together with what the Jewish tradition considers to be the annotation. And you'll see, I'll show you now. So you look at this text, it looks like, for people who don't know Hebrew, I'm sure it doesn't mean very much, okay? But this text is composed of two parts. This is the text, okay? But if you try to read this, it's very difficult. There are many different ways to read it. You don't know where the end of the sentences are. You don't know where to put the accents. You don't know which words are important and not important. And for that, there's this unbelievable system of annotation. Okay? So DNA, which was invented much before that, has a much simpler system of annotation, and that's what I'm going to talk about. Okay. If you look at the genome on a whole, what you see is basically everything is methylated. The whole thing is methylated all over, except for these little regions over here called Cpg Islands. And this is true of every cell of the body in general, overall. Okay? We know that the distribution looks something like this. As I said, everything is methylated. There are some genes that have Cpg island promoters. Okay? The promoter is unmethylated, but the rest of the gene could be methylated. How does this come about? It comes about in the following manner. We start off with our genome basically unmethylated because all the DNA methylation from the gametes is erased. Okay? And then, as the embryo starts developing, the DNA methylation pattern of the DNA is reestablished anew in every individual. We don't inherit this pattern. And it works something like this. Here's a piece of DNA. Here's the paternal DNA, the maternal DNA, and then at the time of implantation in the uterus in mammals, you get a wave of de novo methylation, indiscriminate DNA methylation, probably caused by an increase in the two DNA methylase, de novo methylases of the cell. You get a big increase. Everything undergoes DNA methylation, but Cpg islands are. Am I blocking people here? I'm sorry. I don't know. No, it's okay. Okay. Cpg islands are protected, and they're protected on the basis of a system of transacting factors that interact, which I'm not going to go into, interact with the island and protect the DNA in this region from that process. Okay. Once this is established in implantation, it's no longer necessary every generation to do de novo methylation and protection of the islands. Okay? Rather, the conditions that generated this pattern, basically the factors that generate this pattern go away. Okay? They were in implantation. But then, when the DNA is replicated, there's a system for perpetuating the DNA methylation pattern. You all know this is a very, very simple system. It's based on symmetry, the idea that Cg is symmetric. Okay? If you read Cg in this direction, the opposite strand also has Cg. Every replication cycle, the DNA replicates, okay, by synthesis. That generates a hemimethylated site over here and over here, and there's a special methylase, and together with a number of other collaborative proteins, which then recognize these sites specifically and methylates the opposite strand, thereby perpetuating the original pattern that was developed at the time of implantation. And this goes on, and if you look in an adult, again, this is overall. There are a lot of details in here that we don't see. This is the overall picture. Okay? If you look at Cpg Islands in many, many different tissues of this 70-year-old person, you can see all these islands are blue. Blue means unmethylated, yellow means methylated. They're all unmethylated in every tissue. And basically, the rest of the genome looks like this in every tissue of the body. It's methylated all over. And this pattern, okay, was established in these particular individuals. This pattern was established 70 years previously. Okay? And all of this is remembered. By the way, there's an organism, a yeast, that has been preserved the methylation pattern for 50 million years. It no longer has the de novo methylase. Missing the de novo methylase. But it's been preserved a methylation pattern for 50 million years. Okay. The methylation plays, just overall general methylation probably plays a role in chromatin structure. And every time you replicate, think about this, when you replicate DNA, okay, the DNA polymerase and all its machinery goes plowing through the DNA, it basically disrupts chromatin structure. Following the actual replication, the original chromatin structure has to be maybe not totally reconstructed, but at least partially reconstructed, and probably DNA methylation plays a role in this process. So when the DNA after replication gets repackaged, regions that are unmethylated are put in a more open conformation, and DNA that is methylated is put in a more closed concentration, allowing the perpetuation of chromatin structure as well, not only the DNA methylation. Okay, now what happens after implantation? After implantation, we start development, basically. Okay, so one of the first things that happens is that the organism is no longer pluripotent. And that occurs because a lot of pluripotency genes are turned off. You turn them off, and then the embryo is no longer pluripotent. Okay? And I want to show you, this is at least one gene. This was done in collaboration with Yehudit. This is at least one gene. I'm going to show you how it happens mechanistically. Okay? So this is, let's say, the OCT gene. Okay? It's unmethylated. It was, this could be near the promoter or enhancer, unmethylated. Okay? It has an open chromatin structure, which with acetylation and K4 methylation. Okay? The first thing that happens is that this gene gets turned off by a repressor, a factor that recognizes it and represses RNA synthesis. The first step. Okay. The second step is that G9A recognizes this gene. It's a histone methylase. It brings along with it a histone deacetylase. And together they work, they get rid of the acetylation. There's another enzyme that gets rid of the methylation. And then the G9A causes methylation at the K9 position. That attracts the heterochromatin protein. And now we've generated heterochromatin for this gene. Okay, so we've closed the chromatin. First, turned off for transcription. Then we've closed the chromatin. Okay. The last step in this process is that G9A is able to recruit a de novo methylase. And then it methylates the DNA. That's the last step. Methylation does not turn off transcription. This is the last step. By the way, every histone methylase that we know of has the ability to recruit DNMT. They work together. There's some sort of evolutionary connection between these two things. Okay, so what good is it for? What good is, oh, there's a slide missing, I think. Either that or it's in the wrong place here. Doesn't matter. What good is this? Why do we need the methylation? Okay? So the methylation is for maintaining, for stability. It maintains the oct gene off. Okay? And what I didn't show you is after you do this, after you get this turning off and you get the final methylation, and you try to push the cells back to pluripotency, it won't work. If you use a G9A mutant, okay, that still methylates, but no longer has the set domain, no longer methylates the K9, okay, the chromatin is open now, not closed. It still won't go back. But if you use a mutant that allows you to get the heterochromatin, but doesn't allow you to get the de novo methylation, the cells will go back to their pluripotent state. So it's serving as a way of maintaining inactivity. For those of you who know a little bit about OCT, you'll notice that OCT4 is, there's no instances where it's turned, even in cancer, OCT4 is just not turned on. It's off. Okay? And it'll stay that way forever, right, because of the maintenance mechanism. Okay, what happens after that? After that, we start getting, we get gastrulation, and then we get tissue-specific formation. Okay? So I want to tell you here, DNA, there are changes in DNA methylation. So this is just an example. These are sites that we've looked for in the genome where B cells undergo demethylation. Okay? So all these sites are demethylated in B cells. Okay? They were all methylated at the time of implantation. These sites, not shown here, these sites are all methylated in all the other tissues. So in the formation of B cells, they undergo specific demethylation. Okay? So this is an example of tissue-specific demethylation. Okay? And every tissue in the body undergoes a process like that. We made a knockout in early B cell development of TET2/3, which is the enzyme responsible for demethylation in these cells. Okay? If you make the knockout and then look at the resulting B cell, you still get B cells. You look at the resulting B cells, you'll see that they're now methylated. They haven't undergone demethylation. The knockout worked. What does it do? So here you see a picture of RNA expression from, by the way, all of these sites that you see of demethylation, 95% of them are all enhancers. They're not promoters. And you'll see in a minute that almost all of the genes, many of the tissue-specific genes, actually have Cpg Island promoters that, from the point of view of methylation, are open all the time. Okay, so this shows you an example of what happens with RNA of the genes that are located near these enhancers. And you can see that in the wild type, there's expression from these genes. Okay? But in the knockout, the expression has gone almost completely down. In other words, you don't get expression because of the fact that you haven't gotten demethylation. So the demethylation is required in order to get stable expression in these B cells. Okay, and it has, in terms of the immune system, it has a tremendous amount of, it's very important. So for instance, in the wild type, if you look at the number of germinal centers that you get in response to a particular antigen, you get a lot. In the knockout, you don't get it. So also physiologically, the cells are not working. And this is in general true for the tissues of the body. You have to have specific DNA methylation for the cells to work properly, okay, on a long-term basis. Okay. This demethylation causes an increase. Now, using the knockout and the wild type, we can compare what's going on in these regions, in these enhancer regions. Okay, so if you take a look at accessibility, you see that in the wild type, these enhancer regions are accessible. They're unmethylated in the B cells, right? But in the knockout, the accessibility goes way down. Okay? And the same is true for the binding of specific transcription factors. Okay? Here's an example of a transcription factor that binds to this group of enhancers. And you can see that in the wild type it's high. In the knockout, it's lower. So DNA methylation is necessary. Demethylation is necessary for opening up the region and allowing the binding of factors. Okay? If you're going to take away anything from this talk, is that all of biology is chemistry. Nothing is absolute here. Okay? And I'm going to go over this over and over and over again. Everything is chemistry. Okay. So if we just sort of summarize of what I just said, okay, this is in general true for tissue-specific demethylation. It occurs at genes that have Cpg island promoters. Okay? They have enhancers that are methylated from the time of implantation automatically. Okay? The whole genome got methylated. These enhancers are methylated automatically from the time of implantation. Okay? What happens during differentiation of the specific tissue is that there's a transcription factor that can bind here, even though the DNA is closed, even though there's a closed chromatin conformation, it can bind. Okay? And the binding attracts the demethylation machinery that brings about demethylation. Once there's demethylation, there's a change in chromatin structure, makes it more accessible, and now factors can bind. Notice that any factor can bind now. Doesn't have to be tissue specific. What determined the tissue specificity basically is this, that brought the demethylase and opened this region. Now, many, many factors in the cell, all sorts of ubiquitous factors, can bind here and stimulate the activity of this gene so that it's expressed at a higher level. Okay? So this is the way we think it happens. Now, let's take a look at what happens during development. Okay? So this is a typical picture of what happens during early development. What I want to point out here is that you start off with an epiblast, and somehow you now get branches of development. You got ectoderm, mesoderm, endoderm. This occurs during gastrulation. And each one of these gives rise to a different set of tissues, either endoderm, mesoderm, or ectoderm derived. Okay. So if you now take the whole, you take cells, specific cells in the genome, okay, tissue-specific cells, and you now take their whole methylation pattern, okay, and you throw it into a computer, and you ask the computer to cluster them. Okay? What's similar? What's different? You get the following picture. Okay? So this is just an example of tissues. Okay? But you can see that when we did this, what happened? All of the endoderm clustered together. Okay? All of the mesoderm clustered together. All of the ectoderm clustered together. Right? And then there's a separate cluster for hematopoietic cells. And that's known from development. That's a very, very early branch that is probably derived nearby the mesoderm. Okay? So just by methylation pattern alone, it looks like methylation is directing or having something to do with the generation of the precursors during development. Okay? And the picture we have in our mind, or which I always had in my mind, was something like this. At the time of implantation, you have, before implantation, in the ICM, everything in the genome is unmethylated. This is not real data, this is just an example. Right? And then at the time of implantation, almost the whole genome undergoes de novo methylation, but Cpg islands are preserved. Okay? Cpg islands are preserved in every tissue. The methylation is preserved in every tissue, okay? Whether it's endoderm, mesoderm, or ectoderm. But if you look at specific tissues, you see here's tissue specific for liver. You can see it's methylated in every tissue, but not in the liver. This is unmethylated in colon, but methylated in every other tissue. And every place you see tissue-specific demethylation. And you also see the following. There's some sites that are endoderm-specific. And there are some sites that are mesoderm-specific in general. And there are some sites that are ectoderm-specific. So the way we think this happens, or we'd like to think that this happens, is that early during development, when you make endoderm, ectoderm, and mesoderm, there are demethylation events that occur at that time. And then that gets carried on through all the tissues that are derived from those germ layers. Okay? Okay. You can tell from the way I'm telling you this that that's not true. Okay. Okay. Can you wait till the end? It would be helpful. I'm really fired up. Okay. Let's go on here. Okay. So here's the first surprise. So now we took some of our data, some data from the literature, and we looked what happens when you form at the early germ layers. And this was done by isolating early ectoderm, isolated endoderm, isolating mesoderm. This is a 7.5-day embryos. We took this from a paper from Nature. Okay? And we now take a look at changes between implantation, ectoderm, endoderm, mesoderm. And these are all the places where there's demethylation in these tissues. Ectoderm, endoderm, mesoderm. I'm sorry I don't have the quantities here. There's a good reason for it, but I'm not going to go into it. Okay. So there are some sites that are specific only in ectoderm, some ectoderm, endoderm, a lot that are specific to endoderm. Then there's mesoderm, endoderm, mesoderm specific, and there are some sites that undergo demethylation at all. If you now look in ectodermal, mesodermal, endodermal tissues, this is what you get. Well, methylated. There are a few sites that are unmethylated. I'm exaggerating. But the overall picture, 99% has gotten methylated. So, first of all, you see from the point of view of a reflection of early development, right? A lot of the it just doesn't reflect the early development. Okay? Then you also see that there's something that's interesting here, and that is that you have regions in the genome that are undergoing demethylation. Right? All these events. It's not permanent. It doesn't remain. Okay? It gets remethylated. Okay? So it's very logical that all of the sites here, the genes near these sites, are all involved in what happens at 7.5 days during gastrulation, in those germ layers. Okay? So the genes that are used as early germ layer genes that then get remethylated, and doing motif analysis and RNA analysis, all that's true, okay? I'm not going to go into it. Gets remethylated. Okay. So how does this happen? You know, I've been talking about this for decades, and I always say once a gene gets demethylated, it's always demethylated. Right? It gets preserved, it gets maintained. So the answer is very simple, and that is that there are some places in the genome that start off methylated at implantation, and they have a site, a factor binding site, to bring a factor that will cause demethylation. Okay? And then as that cell remains or perpetuates, okay, that'll remain unmethylated, right, because there's no way for it to get remethylated. There's nothing to remethylate it. But if these sites have multiple regions of binding, one that attracts a factor to demethylate it, and another that can attract a factor to bring the methylation machinery, okay, then you can do this. In other words, the idea of stability is still the same, but now we're introducing here an idea that it's possible also to specifically, just like you specifically demethylate it, you can specifically remethylate it, specific sites, of course. Okay. Now there is a stage right after that. This is at this I call mid-embryo. This we first start seeing, because we don't have earlier samples, we first start seeing it about 10.5 days. Previous one was 7.5 days. Okay? 10.5 days, again, you see, if you go from the germ layer now, okay, from germ layer to demethylated in ectoderm, or ectoderm and endoderm, or only endoderm, or again, mesoderm and endoderm, or everybody, okay? You find these sites that undergo demethylation, and basically they remain unmethylated. Okay, so here you have here sites that unmethylated in ectoderm and endoderm, okay, and they're unmethylated in the adult tissues. This is only unmethylated in endoderm, and it's only unmethylated in endoderm, okay, etc. Okay, the mesoderm, it reflects what happened early during development, just what we expected, but this occurs at a later stage. And the factors that you need to do this are the factors that are expressed at the earlier stage during gastrulation. Okay? Those factors are expressed there, and then they work at this point to cause demethylation that is indeed specific, germ layer specific. And these are basically the sites that gave us that dendrogram that we saw at the beginning. Okay. These are the ones that are specific to germ layer. Okay. Okay. Now, I want to point out that so we've looked at gastrulation, we've looked a little the mid-embryo, we looked at the very, very end of tissue-specific demethylation. Okay? What I'd like to show you now is this process of changing methylation in order to drive development, or stable development. Always keep in mind, what methylation does is stability. Okay? And stable development, okay, and occurs during development. But it also occurs after terminal differentiation. That's what I want to show you now. So here's an assortment of tissues. So every tissue does that. Okay? These are hepatocytes, heart cell, heart muscle, lung, and tissue from the brain. Okay? All of these you see have tissue-specific sites in the adult. 15 stands for 15-week-old mouse. So here, for instance, are genes. These particular genes are specifically undermethylated in the hippocampus and methylated in every other tissue. These specifically in the lung, these specifically in the heart, and these specifically in liver cells. But not all of this occurs during development. If you take a look at birth, the newborn, okay, so there are sites that were demethylated during earlier embryogenesis and remain unmethylated in the hepatocyte, as we would expect. But many sites, almost 40% of the sites, are still methylated at birth. The same thing is true of the heart, even more. Heart muscle, almost all of it was methylated at birth. All of this occurred afterwards. And here too, and in the brain as well. Every tissue you look at looks like this. There are postnatal changes in methylation. Just to give you a glimpse of what this is, okay? And here's an example from the liver. There are many, many sites in the whole genome. If you look at whole genome methylation, there are about 100,000 sites that are, they're all enhancers, that are demethylated in the adult, okay? Methylated in all other tissues, methylated in implantation. Again, these are all sites that were automatically methylated at the time of implantation, remain methylated in all the tissues, but in the liver undergo specific demethylation. Okay? But if you now look, fill in the missing time from birth to 15 weeks, you can see that a lot of it hasn't undergone demethylation, and then undergoes demethylation. The first wave at about 3 weeks, and the next wave at about 6 weeks undergo demethylation. Here's the liver. Okay, and now I've isolated only the ones that undergo demethylation following birth. Okay, so at birth they're methylated, in the adult they're unmethylated. These undergo demethylation early, these undergo demethylation late. There are 2 phases, okay? If we ask what are these sites, okay? We don't have really good proof for this, but it seems very, it's not only reasonable, we have some proof for this. Evidently, these sites over here that undergo demethylation very early are associated with nursing. And the idea is that in the embryo itself, when it's in its mother, right, it's getting food through the blood, basically a glucose diet, right? Comes out, it's presented with this, all of a sudden, this lipid, fat-rich diet, okay? And we think that this fat-rich diet induces an adaptation in the cell, and this occurs through demethylation of these sites. And as you might guess, these have to do with weaning, again, changing the diet from from milk, right, mother's milk, to what we call chow. Okay? And that involves an additional adaptation of further demethylation. So what does this? How does this happen? Right? It happens because through hormones. Okay? And we show that by making a postnatal liver that doesn't have the insulin receptor. So knockout of the insulin receptor, but just postnatally. It occurs, we did it with a virus, postnatal knockout. Just at the time of birth, okay? And we looked at what happens in this knockout. Amazingly, even though this still occurs, the early one, the second stage is completely dependent on insulin. Without the insulin receptor, you don't get the demethylation. So here's a process of demethylation. It's site-specific, it's tissue-specific, it's driven by a hormone Whereas in development, we would have said these things were driven by morphogenes or by factors in the embryo. This is driven by hormones. Another example, if you look in the liver of male and female animals or in man, you see that all of these sites are unmethylated in the liver and of the male, but in the female they're methylated. And this is a postnatal process. It occurs at 3 weeks, and it's driven, the demethylation all of these are methylated at the time of birth, but it's driven in the male by testosterone. And if you give a female testosterone, you can make her demethylated also. Okay? It's all hormone driven. This is an amazing example, again, work that was done together with Yehudi. And this is what we would call, in quotations, real epigenetics. Okay? So if you take a look at muscles and muscle stem cells, okay, all of these sites are methylated in quiescent muscle stem cells, but when the muscle is injured, these stem cells undergo a transformation and become demethylated. These are all different mice, there are a lot of samples for the statistics. And when you undergoes injury, all of these sites undergo demethylation. Okay? And these cells now remain. So if you now take a look 2 or 3 months later, this mice still have stem cells, but they're no longer, and they express, their expression is just like quiescent stem cells. But epigenetically, they have all these sites open now. And epigenetically, they preserve this now sort of activated state. Okay. Interestingly, you injure one muscle, you get the same thing in other muscles. Okay? And we know that the factor that does this, the factor is hepatic growth factor. So when you injure one muscle, it through a little biochemical series releases an hepatic growth factor, which affects all. It's a hormone. It affects all the muscles in the body. They all undergo. So you injure the mouse once in a muscle, he now has all his muscles in a more activated state. They're epigenetically ready. And even more amazingly, and this the women will be very happy with, in following pregnancy, pregnancy itself also causes, in the muscles of the body, causes this activation, this epigenetic demethylation. How it works we don't know, but again, it's something hormonal. Okay. Okay, so postnatal changes in methylation look something like this. You have a hormone. The hormone interacts with the cell, okay? And it brings about the activation of the transcription factor, which now recognizes certain genes, certain enhancers, okay? That brings the demethylation machinery. The site undergoes demethylation, the chromatin changes, and then you get acetylation of histones and interaction with many proteins from many factors from the cell, turning on these genes, but the genes don't necessarily remain on. Transcription has its own regulation. This is the epigenetic regulation. Okay. And so I just want to show you that what I showed you about what happens during development, that we get demethylation and then remethylation. It occurs in other places as well. So here's an example, for instance, of we isolated hepatoblast from 11.5-day embryos. Okay, so these are the precursors of hepatocytes and cholangiocytes. Okay. And we've looked at the sites that are, quote, tissue-specific demethylated in hepatoblast at 11 days. Okay? In other words, these sites are methylated in all other tissues. They're methylated at the time of implantation. In hepatoblast, they undergo specific demethylation. Okay? Now let's look at these sites in hepatocytes and cholangiocytes. Okay? Almost all of them undergo de novo methylation. In other words, these are sites, this group undergoes de novo methylation early in everyone, and this group, some of them undergo de novo methylation in the hepatocytes, but not in the cholangiocytes, and some of them undergo de novo methylation in the cholangiocytes, but not in the hepatocytes. Okay? But almost everything undergoes de novo methylation. By the way, after this, there's, in the process of making these tissues, there's additional tissue-specific demethylation after the hepatoblast. But this, everything that happens in the hepatoblast is sort of, once you've done it, it's a race. You needed it for that stage of development. You don't longer need it anymore. Okay? Another example, here's the blood system, hematopoietic system. You all recognize it. There's a stem cell. It gets multi-potential cells and then divides down into lymphatic and myoblastic, lymphoblastic and myeloblastic, and generates all the different cells in the lymphoid and myeloid systems. Okay? If you take all of these sites, so first of all, if you take a look at hematopoietic stem cells, all of these sites are tissue-specific demethylated and just in the stem cells. And these are other tissues, it's all methylated. But now look what happens is all of the cells in this system, all of these cells, and these cells, all of these, a large number of them undergo de novo methylation. All of these enhancers, okay, they are opened up in the stem cell and then closed before the differentiation of this into individual cell types. Okay? A large number of them also remain. Okay, so I think I've made my point. Let me just, let's see what's next. It happens in the muscle as well, and we don't have to do that. So what I'd like to do is, I'm going to skip a lot of what I planned, as usually happens to me, but that's okay. I just want to go to the very end. What did I do here? I did something wrong, of course. Oh, okay, the most important part. And this is not right, but okay, no, it doesn't matter. So I'd like to thank the people who were, it's okay, who were responsible. And the basic hero for the development story is Oprah, working together with Howie, who does the data analysis, bioinformatics. I'd like to thank Shari for her work, Shari Olansky, and Safi, who did a great deal on tissue-specific and postnatal demethylation. And Yossi Buganim's lab, especially Ahmed, who I didn't get to discuss his work, but he's part of this story. And Yehudi, as Yehudi already pointed out, and I'm going to point out again, we have a long-standing, wonderful collaboration, which is just not only enriched the science, but enriched our lives as well. Tal did a lot of the work, the work on the muscle, and then part of the work was done by Ben Sanger in Philadelphia together with his student Jason. And I thank you very much.
Thank you, Howard, for this really fascinating epigenetic tour. So we'll have some few minutes for questions before we end. So here is a question there.
If you go back to the slide where you showed the hematopoietic stem cells and plasma,
Yeah. I have a feeling that I'm going to, oh, maybe we'll get it here. Luckily, okay.
so you said the left methylated column with the yellow is compared to other differentiated species, right?
Yeah, this
So-
one.
Yeah, and the column next to it is the hematopoietic stem. Yeah?
This is the stem cells.
Yeah, so all the genes that undergo remethylation in the hematopoietic progeny, could you infer from that? Because I see it's the same as in other differentiated cells. Could you infer from that genes that maybe are necessary for stem net?
Is it what?
Genes that are necessary for stem, like stem cells?
Yeah, that's, and if you take a look, if you do Go analysis and motif analysis of these particular genes, that's what you get.
Alright.
With a tremendous P, minus 100, something like that.
And can you do that, like, is it also correct for other stem cells, for other tissues?
Yeah, the same thing is true for other stem cells. And we think, we haven't really looked into this, we think a lot of the ones that undergo in this stem cell also undergo demethylation in other stem cell systems as well.
Alright.
It's interesting. Now, according to this, it looks like everything that's unmethylated in the hematopoietic stem cell, these
Yeah.
remain unmethylated, and that's true, but that's only a fraction of what's unmethylated in these other tissues. There are more tissue-specific
Yeah.
incidents of demethylation that occur from here to here, right? From here to here, and from here to here. Yep, you're the boss.
Okay, thank you for asking there.
I was wondering if you know why DNA
You have to speak up because,
I was wondering if you know why DNA methylation is so important in mammals, as you've shown, but C. elegans or Drosophila that they don't have any methylation, they
right.
still have good development.
Yeah. So I didn't get a chance to show it, but you basically what drives development of factors. That's what drives development. But methylation provides its stability. You can do everything with the factors. That's the way it works. Because differentiation is specific, it has to be driven by factors. Okay? No other way. But the methylation provides its stability. You have to look at it that way.
Yeah, that was really interesting. So I still have a conceptual issue with the observations that you made that
Welcome to the club.
I hope you could learn. So there's gastrulation specific, transcription factors that become active and demethylate certain regions and so on. It makes a lot of sense. But then you show that many of those sites become methylated again later on. But there's also going to be factors that are needed to give that phenotype to that specific lineages that are generated during germ layer specification. So, take a muscle cell specific transcription factor. You want to keep that on also later on. So, and if those loci become methylated again, is that correlated to shutting down those genes again? Because we always assume that the expression is correlated with the phenotype.
Yeah.
So I'm puzzled by that observation.
I was also very surprised by this, right, that so many of the demethylation events in the very early germ layers, right, then get closed up.
Yeah.
Right? There should be ones that stay. There are a few.
Okay, so there's
But
exceptions.
number-wise,
Yeah.
it's overwhelming.
So,
And what you're saying is true, I think, that there are sites that are required, genes that are required, and all of the Go analysis and motif analysis really confirms this, 1,000-fold over. And that they are needed for something that occurs at the time of gastrulation. It's very important for gastrulation. It's not necessary afterwards, evidently. I'm also surprised.
Thank you very, very much, Howard. Thank you, all the speakers and all the audience for this really, I think, brainstorming discussion and this whole meeting.
Okay, wonderful. This was a inspiring session and a very nice ending to the scientific part of the school. Now we have 2 more exciting days, but before, one of the most important sessions of the school is the poster session. And to lure you into the tent, the sandwiches are there. We're going to have 2 30-minute poster sessions, the one starting now from 1 until 1:30, and the other one from 1:30 to 2. Now, you still don't know in which session you're presenting, but this is Eitan, and Eitan maybe he will say a few words, but he will also be there at the tent, so ask him if where your poster is. You want to say something?
Okay, so there are 2 sessions of 15 posters. You'll see the sign at the entrance to which poster session you belong. There are 24 monitors there, but only the first 15 are allocated for us for the school posters. The rest are for tomorrow. After the poster session, those who registered to the tour, we have a bus waiting for us outside. But before that, please come back here or to the lobby downstairs. There are water bottles to take with you. And then at 6 o'clock, those who are not coming, you're also invited to the 6 o'clock dinner at the Ima restaurant, which is, the Israelis probably know, it's very famous in the street of the Shuk in Agrippas on the corner of the main road there. And that's the plan for today. So let's enjoy the poster session. Thank you very much.